Hello, I'm Dr. Ben Newman. I study coronaviruses, and this is Ask Dr. Ben, where I try to answer your questions. All right, next question is from, shoot, I never did figure out, is it Cheryl or Cheryl? Yeah, guess it depends where you're from. Anyway, welcome. Yeah. Okay, the question is, are viral vector vaccines, such as Johnson & Johnson, and that's, which is the American version, or AstraZeneca, or whatever, yeah, there are several, a more conservative approach for vaccinating children after trials are completed successfully than mRNA vaccines, since viral vector vaccines have been around since the 70s? Fine. All right. Viral vector vaccines, yes, do have an older history. The thing is, with each viral vector vaccine, you essentially have to try it. You're not sure that it'll work, and you have to tinker around. You play around with different virus shells, and there are many different opinions on which outside of a virus is going to make the best version of this. We've settled on adenoviruses because I think they grow really well. This is a vaccine that you essentially have to farm up, and like it's basically like milk and cells every day with your big bucket of vaccine that you're trying to get out. And yeah, it's got all the uh, all the wonder and really hard work and frustration just out of nowhere that I would imagine farming carries. Yeah, as a non-farmer. Yeah. But yeah, I think the technology does go back, or at least the idea goes back a little bit earlier. The ideas for nucleic acid vaccines, though, come from the 1980s. So they're both pretty old ideas that have been around and not terribly effective, but somewhat effective for a long time. I don't think in science that an older technology is necessarily better. I mean, leeches have been around as a treatment for whatever ails you for a long time, as has castor oil, as has snake oil, for that matter. And I don't think being older makes them better. Yeah, in some cases it makes them worse, but in other cases it's just compare how well they do at a particular time. And yeah, I would say that's a good way to analyze these. So in terms of the vaccines, the RNA and the vectored vaccines seem to work at about the same frequency, and they seem to do pretty well in just about everybody who gets it. The Johnson & Johnson vaccine, if you give 2 doses, works about as well as the mRNA vaccines if you give 2 doses. I know it's advertised as a 1 dose vaccine, but in the trials, in the things that they published, they tested it out as 1 dose, 2 dose, high dose, low dose, yeah, whatever, 1 fish, 2 fish, and yeah, they decided that was a workable thing. Ultimately, when you do a clinical trial, you just pick something that you think is going to work, and you do it as hard as you can. And if it works, then great. Your company makes some money, and you've contributed to society. And if you picked wrong, well, too bad, buddy. Yeah, you got to start over, or you're not going to get to the finish line on this one. Yeah, that's the idea. So, in terms of, yeah, I guess it's conservative because it's older, but I don't think that's what I would look for in a vaccine. So all the stuff that you hear about people saying that mRNA vaccines are too new and haven't been tried, that's bunk. Yeah. These things have been tried in humans since the mid 2000s. I think 2008 was the first publication that I can remember, and they're fine. As are vectored vaccines in general, although there's lots of different vectors, and you have different good and bad sides to each vector. There are a lot more vectors out there than just adenovirus that people use for different purposes. Now, some of these vectors would potentially change DNA and leave more of a permanent gene inside of a person. Those would be good for genetic conditions where you don't want to be treating every day, like some clotting disorders, for example. You have other vaccines that are designed to generate a little bit more of a response, but they're still vectored vaccines. I'm thinking like an alpha virus vectored vaccine or something like that. And those can be fine. Yeah, there are as many vectored vaccines as there are groups of viruses, I believe, or ideas for them anyway. And yeah, you could honestly make a coronavirus vectored vaccine. A lot of people have published these things. And they're okay. It's just a matter of what you can make enough of quick enough that you can get it out to enough people. Because for all of these vaccines, it takes a lot to get enough of the vaccine together so it works. The nice thing with mRNA is that if you need more, you just add more ingredients to your giant bathtub that you're stirring with a big metal spoon, I imagine. And yeah, it's essentially almost a chemical synthesis process. You need, 2 ounces of this, 4 ounces of that, and a little sprinkle of RNA on the top. And yeah, stir it up, and you've got yourself a scalable vaccine. And I think the other thing that we saw is that you can make a vaccine faster with mRNA because you don't have to farm up all the little components the way you have to do with a vectored vaccine. The thing with growing vaccines like this, and I'm sitting here at Texas A&M, I guess I'm an Aggie, I'm going to argue against farming just this once, yeah, but only for vaccines. We should not be farming our vaccines. That's yeah. If it's the only way, fine. But if there is another way, like mRNA, that just seems so much more efficient. And problems like pandemics are very time sensitive. You want to be on the spot faster with a solution. You can wait. You can do many more years of testing, but how many more people die, and how much does it just slam everything to a grinding halt over and over again as we have surges? No. Faster and more effective is definitely the way out. And yeah, it turns out that the vaccines are safe as well. So I don't think conservative is the way I would choose to, conservative, or I don't know, liberal, yeah, whatever, or maybe newfangled, yeah, is the way I would choose to view vaccines. I would look for the ones that give the strongest response, longest lasting immune response. I would look at different kinds of cells. Is one better at educating a particular set of T cells that might be more helpful, things like a Th1 or a Th17? Do you have one that's better at producing really good memory B cells? Do you have one that makes a lot of neutralizing antibody? Do you have one that's going to make antibody that's going to be resistant if the virus changes a little bit? I think all of these are good concerns. And yeah, with the RNA vaccines, it's just a faster, shorter path to address those concerns than it is with these farmable little vectored vaccines. So that's the side I'm on of this one. And I don't think you have to worry about that. In particular, no. And these have been given to so many millions of people now safely that I think that gives me a lot of confidence personally. So thanks very much. This has been Ask Dr. Ben. Hope that answered at least some part of your question. And yeah, I hope you stay safe out there.